Good morning, welcome to this BioRails demonstration. I'm going to show you the features of the BioRails electronic lab notebook and also take you through how they're used in the context of a request fulfillment workflow focusing on the needs of a DMPK CRO laboratory. So what we're going to start with is a quick presentation of the BioRails ELN features. BioRails system is made up of a number of different entities. Each entity has a folder system. That folder system is used to track the version control contents for each experiment and other entities in the system. The content within the folder can be directly printable to PDF, which can be used for archival processes. It can also be used to exchange protected information between the CRO and their customers, which can be cross-checked for IP protection purposes. It uses a role-based access control mechanism, and there are state flows to control the workflow, including the signature process. All these things can be accessed from the business objects such as request and experiment, which configure the appearance of the folder and the content that's allowable within that folder. Here's an example of an experiment for an in vivo PK study in which we can see details of the structured data and a morphit binder which has been embedded in the experiment. All of the content within the folder is version controlled. So this will version all of the content of the folder, not only the morphit document and its content, but also any extra information that you've added to the folder. This builds a very effective content management system which allows you to attach any files and other assets that you want to keep with the experiment. These can include Office documents such as Word, Excel, morphit files which can interact with the structured data in BioRails, PDF files, as well as raw data files if you want to archive those with the experiment. If you attach image files such as cell cultures or histology images, they can also be annotated and notes can be added to fulfill and write up the experiment as you would expect with any electronic lab notebook. So we can add instructions of the protocol, we can add details of what actually was conducted during the experiment as well as our conclusions. We can also reorder the contents of the experiment in order to control the format and printout. So here is a section of the morphit template. I've added a histology image which I can then annotate by adding annotations to highlight different areas of histology and mark those with different annotations. All of the content of the folder is sectioned. The third aspect of the BioRails electronic lab notebook, which differentiates it from many other notebooks, is that it's based also not just on unstructured information but also on structured data. It uses aspects of the BioRails DM system to be able to create process versions for recording structured information and have associated experiment templates. These allow you to add integrated data analysis for things like curve fitting, statistical summaries, charting and graphing of data. All of the data that's captured in the structured data elements of the BioRails ELN are validated against their source data systems. So this means that you can control the master data such as dictionaries of compounds, batches, dictionaries of species and other elements that are within the experiment recorded, which makes sure that you capture data of high quality at the point of data capture. In this example here we can see that we're searching across a number of different experiments where we're capturing the structured data within those experiments. So here we can search by EC50 and R² values and bring back the graphical information from the data analysis. Any electronic lab notebook, of course, is also responsible for protecting the intellectual property that's generated during experimentation. And BioRails contains features of electronic signatures in order to protect that intellectual information. There is a signature record which provides a permanent record of the data. It's tamper-proof, providing MD5 checksums of all the content, so that if someone goes in and modifies that data at the back end, the system will invalidate the signatures. This provides everything you need for a defensible document you can take to court, including support for the signature workflow, so it can be signed by author and cross-signed by witnesses as well. There's also support in there for managing that signature and witness workflow, with automatic emails that are sent out to the witnesses that informs that they need to witness an experiment, as well as reports in the system which quickly allow you to review all of those experiments that you've been asked to witness and go through the witnessing process. So in this example here, I'm signing a version of my experiment as well as selecting a witness. The witness would then be sent an email. They click on the email link, it takes them to the signatures page. They can see which version of the document it is. They can print it to PDF and they can countersign the document. And that builds up the defensible record in the intellectual property that can be used to defend the intellectual property generation in court. So that gives you an overview of all of the different elements of the Biology ELN. So it allows you to build up content within a folder. It allows you to manage that content in terms of version control, add additional files, images, annotate them, and review the data. It also allows you then to capture structured data within the same experiment to enable searching, and also then to sign off on that content. So what does that mean? That means that your intellectual property is protected and it's built into the scientist's daily workflow. The data is captured in experiments and can be analysed to create knowledge, and of course the structured data is also captured to allow you to be able to search and mine that information as well. And all of your biological research can be managed in this single ELN system. Once the data is built up in the system, you can then analyze and use that data as an asset to your organization. So it allows you to use reporting tools to be able to search and evaluate the data. It allows you to use a variety of different reporting tools. So here's a couple of examples. It also allows you to use that structured data to improve the quality of your experimentation. So here is an example where we're using previously captured results for controls and standards to then analyze whether our current run is within statistical variance of the overall data. So this is using a Morphit template to query that structured data and then plot out basically the performance of verapamil standard in this assay over all historic results, and I can immediately see if the current result is within a certain number of standard deviations of the mean of all the previous experiments. So you can see some of these have fallen out of the range, and we could choose not to publish that data on that basis. The system is very flexible. It allows you to create lots of different templates for different experiment types. So we can use this for in vitro experimentation. Here was an in vivo example for a tumor volume study, a xenograft study, in which case which we're analysing the performance of different animals amongst different control groups. All of that data is stored in the underlying structured database, so it can be used for routine in vitro work, it can be used for exploratory in vivo, and all different flavours in between. And all of the data is structured in a structured fashion, so you can search and mine the data afterwards. Of course, all the data, including the intellectual property, is all archivable. So it gives you a defensible document which contains details of all the experiment and the results recorded, your observations, and of course importantly details of the intellectual property protection. So you can see here it's you've got a hash code which makes sure that the data hasn't been tampered with, and also details of the signature are in there that can be used as a defensible document within court and cross-referenced by all of these hash codes. You can also drill down into details of the underlying documents, which are also checksummed. So we've got multiple records, so to make sure you can identify each individual document that maybe were tampered with. Here's an example of what you would get if you had gone in and modified the data through the back end. So we go and analyze the signature for this particular experiment. You can see it's failed validation. It will tell you basically that its current checksum doesn't match that of the signature, which indicates that this data has been tampered with. So that means you've got archivable, defensible documents and also protection of the intellectual property from tampering. All of this again means that we have good intellectual property protection within a system which manages your workflow, allows you to record all of your structured and unstructured information, which allows you maximal use and keep the value of that data and use it for all of your biological research. So now I'm going to switch through and go and show you the context of this within a demonstration system as part of the request fulfillment workflow. So this is a BioRails system. I've set this one up to demonstrate a system used for a contract research organization. So I'm going to log in as someone called Walter. Walter is a project leader. He could be a business development person within the CRO recording customer requests, or indeed you can also let customers into the system, allow them to enter their own requests into the system. So what we're going to do is make a new request. Requests can be based on lots of different things, but we're going to make this one a lot of compound to be searched. So that lot will be extracted from the chemistry registration system, and I'm going to say it's for customer B. Click next. I'm then going to add in a list of lots which can be looked up from the database. So in this case we've got a series of samples, and also you can see in the database I have my controls and standards as well, or I could paste in a list, or I can import them for a file as well. I can then select the services that I want to submit these compounds to. So you can do that by a whole panel of assays. You can also set up a workflow which will allow you to run a series of assays and decide which other service would be run consequently after that first set of services. In this particular example I'm going to request some DMPK services. So I'm going to ask for a microsome stability, a brain binding, and also an in vivo PK assessment for my compounds. Next, the system's going to basically ask me for any details of the request. So you can see I've got three different services here. The first one is a mouse microsome study. So you can see it's fixed at mouse. I'm not allowed to change it. I can add a comment against the compounds if I want. So I could say that one's input. I could say that one is important. In this second example here, it's asking me to say which species I would like. So I'm going to look up and I'm going to say I want a dog microsome, and I'm going to want this for all these compounds. I haven't got a comment to make. The final one then is an in vivo PK. Here I can specify which type of in vivo study I'd like. So I'd like a bioavailability study, and I want that in rats, and if I want to I could specify also the strain of rat I want to use as well. And also I can specify which gender of rat. So I'm going to use male rats, and I'm going to copy these down for all of the compounds in my request. Click next. Then what it does a check. It says, have you asked for that same compound to be tested before in this service? Because if you have, you may not want to ask for it again. In this case it's a demonstration system, so I had asked for it before, but I could click on that link and go and see which experiments have already been conducted. So let's say I'll have all of those again, and then I'm going to confirm that request into the system. So here I get my request dashboard. You can see they're pending in all those different assays. Walter can then review all of the requests that he's made. Up here you can view them all, or you can also look down at the item level as well. You can see some of these are color coded because there is a turnaround time for that particular assay, and it's color coded as it approaches its deadline. So you can see these are brand new, they're highlighted in blue. These two samples here are past their deadline date, so they've gone red. And you can control those turnaround times or service levels for that particular assay. So that's my requester done. So I'm going to log out as the requester now. And now I'm going to log in as someone from the test lab. So Bill is running the DMPK laboratory. You can see he's actually running the plasma protein binding, the microsome stability, and also the in vivo PK assays themselves. But he can get an overview for the lab as well by clicking on the View Services link there. And here you can see I've got a number of different assays being run in this laboratory. They range from blood plasma ratio, in vivo PK assessment, and there's also a plain bioanalysis tissue sample service as well. For each one of the services I can see which scientist is actually managing those queues, and also see how many pending samples there are to be done. So you can see some of these services have got lots of requests still pending. There's 16 ongoing in this example here. So what I want to do now is I want to actually schedule a microsome stability study. So I simply click on the link. That takes me to the queue for this assay. I can then review all of the requests coming in. I could have a look at all the teams that have sent me materials. I could also look for specific customers in here. And then let's say this one is for chemistry. So I only want the chemistry requests. I'm going to select all of those. I'm going to put them in the cart. And you can see the next thing it's done actually is it's done a stock check to see if there are any tubes in the tube store that match this request. Now, if you're using the BioRails inventory module, then it can check inside of that. If you're using another inventory system, such as Tissue Mosaic or an internal one, then we can integrate those systems to allow that stock checking process to happen, either here within the assay queue, or also can happen within the request process, so that people don't submit requests for things that are out of stock. In this case, if I wanted to, I could go ahead then and order those tubes to be delivered to the lab. For my example here, I'm going to select only those with stock. So you see the bottom three didn't have enough compound material to be tested, and then I'm going to create a task from those. I then go through, you can see it's automatically generated an experiment identifier, and I can pick which protocol I want to run for this particular experiment. So I'll keep it to me, but if I wanted to, I could give it to somebody else in my group. I can also give them some basic scheduling information. So it's Friday today, so I'd like him to start that on Monday, and he should be able to finish it by Friday. So we'll click Save, and that will create that experiment. So here's my experiment. This experiment is a mixture of structured information, which, as we described earlier on the electronic lab notebook. It's also got a template embedded for actually processing the data. And if I want to, I can add other things to it. So for example, I might want to add in here a description of my conclusions, or I might want to put in something about the protocol itself. These sections can be automatically filled out with the template for the experiment, so that you always get a running copy of the protocol, or you can add them dynamically, which we're doing in this case. So we're going to say, actually, let's make it that. Step one would be to plate compounds, incubate microsomes, and then obviously more steps that will be added. So that's added to the experiment. You can see it's added to the end automatically. If you want to reorder it, you just go to the content view and you can drag and drop it so that it moves up to the top of the experiment. So here now you can see there's my protocol above here. So what we're going to do now is run this experiment. And in this example here, I'm going to run it using a Morphit template, which was embedded when I created the study. So I'm going to click on the link. That will then fire up Morphit and download the information that's already been filled out with this experiment, such as the list of samples to be tested. So here's our template. Then all I need to do basically is work through the instruction sheet. So we'll start off by going to settings. You can see it already knows which experiment from BioRails, and it's got a date in here. And you can see it's got the protein concentration. It's also got a minimum area threshold, which is used to automatically knock data out. So I'm going to import my file. In this example here, this is an analyst file. So anybody who does mass spec will recognize this. So you've got all of your data in here. What I've done previously is to create something called an import definition, which Morphet allows you to basically read any sort of file with a variety of different patterns in it, and you go through an import definition to teach the system how to read the file. So we're going to read that data in, and it's going to calculate out all of its different values. The first thing it does is take you to a primary data review table. So you can see some of these have been automatically knocked out. So if we look at the formula here, you can see that if the basically if the sample response red is less than the minimum area, that's going to knock it out. So you're knocking out certain values are below the threshold. That threshold value was in the settings. Then all I need to do is go through my protocol here. So we're going to look at batch information first. So these are the samples that we're bringing in. We can look at our calibrations if there are any. I don't think this particular one has any calibrations, but if there were, they'd be displayed. Then I can look at the actual analysis. So here you can control so you might want to drill into one of these. So here we have a plot of the percent remaining against the time points, and we're using a linear fit to the data, and we're also using a one-phase exponential data to fit to the same data as well. Obviously you have just the percent remaining, not the log percent remaining. These graphs are all interactive, so if you did think there was an extraneous data point that you wanted to knock out, you just double click on the point and you can see the R squared is being refitted and replotted. So you get some indication of the quality of fit, and then you've got your R squared values which are calculated, your K values, standard errors, your half-life, and your area under the curve to infinity. So that's all of your results calculated there. You can obviously go and review the curves for all of the compounds in the set. If I want them all to be the same size, I just need to, and then you can just scroll through all of the curves. We've also categorized the results as well. So for each compound you can see I've got my standards and controls there, and then the individual compounds. We can go and review all of those. And on the right here I've got my calculated Clint values, the low, the high, the scaled clearance according to scaling factors depending on the species, and predicted clearances as well, and of course my extraction ratio at the end. If I want to, I can add some comments in here to comment on particular compounds within the set. Then finally I've got my conclusions. So we've just summarized and categorized the results depending on whether they're high and low or medium, and then I've got all my data in here. So you can see there might be a problem with this particular compound here that I could go and examine. The final step then is to post the data back to BioRails. So that will send all of the structured data out of the Morphet document, including the graphs, charts and data, and store them into the experiment in BioRails. So that's the upload complete. So if I go back to BioRails itself and refresh the page, you're going to see all of my data's back here. So I've got the charts and graphs for the experiment. I've got the individual data values as well, all on the same page here. So there's the graphs loaded. Of course at this point I might want to add my conclusions to the study. So I'll add a conclusion and we'll say okay, and we'll just add that conclusion to it. So here we are, we've got all our data stored inside the system, including the updated Morphet template with the data in. but all the structured data has been extended. So now basically if I move this to signed, it will ask me for electronic signature, and I can sign it and then send it for a witness in here. Okay, so in this case I'm not gonna sign this example. That's how you would sign off the experiment, and then of course when it's ready for everyone to see, I'll move it to published, and then that releases the data for other people to be able to go and make decisions on. And those signature events we can control on whether it's published or signed. So that's completed my experiment now. So what can I do with the results? I want to show you how you can search and mine that data. So we go into the queries here now. You can see I've got, first of all, I can search for all the microsome experiments previously run. I can see their graphs and charts. I could go and look up an experiment as well. I can also go and search for the results. So if I was looking for a particular compound, then I can type in the batch code here and go and find that compound. I can also search the results in here by threshold, so I might want to see things that are less than 0. That would pull back all the values. Obviously I'm running the same thing multiple times with different with the same data sets in this example. So that shows you how to use the electronic lab notebook, how to use Morphit for data analysis, and also then how to sign and witness the content and then share it out as well. All of the data in these reports is exportable out to Excel, so, and you can control how many rows are allowed to be extracted. So if we open that one, here's all of the structured data that's been extracted from the system. So that concludes this demonstration. Thank you for your time.